It's time now to journey into the nucleoid of Norbert's cell and to look at this, what looks like a large mass of spaghetti, and to qualify that as actually being a non-membrane bound but contained region or discrete region in which there is stored the genetic material of the bacterial and archaeal cell. And it contains RNA and protein, but primarily it is comprised of the DNA of the cell. And for most bacteria and archaea, that is to say that it's one single chromosome. Catch that? I've got a little demo here in the form of a TheraBand. I bet some of you have one of these at home. If we pictured that this was in fact the fully strung out, elongated chromosome of a bacterial cell, one quick note is that in the cell, once it's unpackaged, this chromosome is actually a circle. So if we could picture this as being the chromosome and being a circle like that, that is what it would be if we were to take away all of the packaging from it. And then if we were to cut it, then we could cut it into one long kind of linear chain. And guess what? This, if we completely strung it out, would be 1,000 times the length of the cell, 1,000 times. I mean, this particular TheraBand is like maybe double the length of Norbert down there. So that's a lot, right? That's a lot of DNA. And it's got to package into less than 10% of the volume of the cell. So you're thinking, how in the world does all of that get scrunched in and packaged in there? So in fact, it does so via something called supercoiling. So I'm going to see if I can supercoil this. Literally, I'm turning at both ends in opposite directions to show you how one of the layers of packaging takes place. The most important packaging in a bacterial cell is via supercoiling. So you can see as I twist and turn there, I'm introducing the supercoiling that's going to take place. And if my TheraBand behaves, you guys are so laughing at me at home. We can see that the supercoiling is eventually going to get to the point that it's going to help to highly package. And you can see how that sort of spaghetti-like structure starts to form. So this is called condensation. So you can see how we start to get to smaller and smaller volumes that this nucleic acid would take up. So that single circular chromosome is packaged down to such a point that it only takes up 10% of the volume of the cell. So the nucleoid, that gel-like mass at the center of the cell, is comprised primarily of DNA, the cell's chromosome. But it also is packaged partly with protein, and we of course will have RNA at any given moment, partly because of expression of the DNA. That is, DNA is converted to RNA, which is converted to protein. That's the whole central dogma of gene transfer, right? So this is the nucleoid in a nutshell. Gel-like mass of DNA, RNA, and protein, primarily DNA, more 60% or more of DNA, and it is packaged by supercoiling. So it's a single circular supercoiled piece of double-stranded DNA. Now it would be a couple of things. We say that bacterial and archaeal cells do not have a membrane-bound nucleus, and that's one of the primary things that separates them from the eukaryotic cells. However, there are some types of planktomycetes, for example, that are bacteria that have a membrane-bound nucleus. Likewise, there are some kinds of bacteria, like streptomyces, that have linear chromosomes instead of the circular chromosomes. However, as a generalization, we often will draw a bacterial cell like I have done here with a chromosome that's laid flat onto the page in its circular form. It's double-stranded DNA, so you can see the two strands. This is not a really accurate description of it because we know that it would be packaged partly via supercoiling, but it does show us that this is the single circular chromosome. Now, this is not the only DNA in the bacterial cell. So whereas that, and we're going to just get Norbert again. You guys are so laughing at me. So we've got our nucleoid, right, with the single circular chromosome. Here in Norbert, it's shown as a fully packaged, supercoiled and all. But notice too that we've also got these little pieces of DNA these little pieces of DNA outside of the nucleoid, right? They're not a part of the nucleoid. They're outside of it. They're in the cytosol. These are called plasmids. Plasmids are crazy. Plasmids are much more common in bacteria than in any other form of life. They are also found in archaea, but not to such an extent. And they're observed in some eukaryotic microorganisms, some eukaryotic life, but they're not particularly common. So in bacteria, plasmids are very common. They're outside of the chromosome. In fact, we call them extrachromosomal, outside of the chromosome. They multiply independently of the chromosome. So that is, they have on them their own origin of replication, sometimes called an OREC. So they multiply without the chromosome, and guess what? They're not actually needed by the cell in order to live. So they're not required for replication and growth. However, guess what? They do make the cell into a superhero, because plasmids often contain extra genes that give the cell a selective advantage. For example, they might give the cell the ability to thrive in the presence of heavy metals. Or, even crazier, they might give the cell the advantage to thrive in the presence of antiseptics or disinfectants or antibiotics. Crazy. So most plasmids are circular pieces of double-stranded DNA. However, some are linear. You can see why a cell would prefer them to be circular, because then they don't have the ends that might get chewed up by nucleases, right? Pac-Man enzymes that chew from the ends of DNA. So some linear plasmids have been observed, but circular plasmids are more common. They're not required for growth, but they do give the cell a selective advantage. For example, antibiotic resistance. And this is where things get kind of nightmarish. And I'm going to grab my tablet and I'm going to show you something here. What if this cell that contains these plasmids, what if one of these plasmids, say this one, is a nightmare plasmid, this plasmid maybe is an R plasmid, stands for resistance. And let's say that this plasmid also is self-transmissible. What that means is that it can encode for this bacterial cell's ability to make a pilus. And this pilus is often termed a sex pilus. It's a structure that allows this bacterial cell to link to a second bacterial cell called the recipient bacterial cell. So say over here we've got another bacterial cell and it gets connected via this sex pilus, and then this plasmid can get transferred across. So this plasmid could get literally sent over to this recipient cell. And what if this carried the gene for resistance to tetracycline or ampicillin or amoxicillin or all of the above? Possibly it would. And then this is transferring resistance from one cell to another. This is a process called conjugation. Conjugation is crazy because over the course of digestion, in just hours, an antibiotic resistant bacterium can conjugate with your normal flora and cause them to become antibiotic resistant. So we can see this transmission of antibiotic resistance genes being incredibly pervasive. In fact, I have this awesome poster in the lab, and you guys should go look at it. It's on the back of the moving chalkboard, and it's got this great cartoon where there's this little bacterial cell like in like the trench coat in the alleyway, and there's another bacterial cell walking by, and he's just like, psst, hey, you want an antibiotic resistant plasmid? And he's selling the resistance. And this is so true, right? It's a bit of a nightmare story in which these plasmids can really get around and do get around to many recipient cells, and then our recipient cells gain antibiotic resistance. So this is what can happen with conjugation. Now, lastly, I want to make note that plasmids are another tool in a research lab. Soon in lab, you all will be taking a plasmid that contains the gene that allows a cell to glow. That is literally fluoresce. We can put it on a UV light once it has this plasmid and it will glow. So the use of plasmids in the lab is very, very common because we're always wanting to give bacteria new genes. We're wanting to make them express new genes. What's kind of weird is that we usually actually also include an antibiotic resistance gene on these plasmids, so that when a bacterium gets this new plasmid, we can actually find out which bacteria have it by looking for their ability to live in the presence of antibiotics. So these are often used by researchers to clone genes. That's what cloning means. Cloning means to make a replicate copy. So when we put in a gene on a plasmid and we ask bacteria to make more and more copies of it, we are cloning genes. That is what we're doing. You guys are going to be cloning, and you can call home and tell your parents, guess what? In microlab, I'm going to be cloning. And then they'll freak out, and you can explain that that means to make a duplicate copy, a replicate copy of a gene. Okay. So this being said, I want to show you some pictures. These are varyingly complicated. So let's begin up in the upper left with the less complicated view. Notice that this has the origin of replication. That means that this plasmid is able to self-replicate. It's able to make copies of itself. This one also is going to have that. So all plasmids are able to be replicated. Now, the ampicillin resistance gene over here and the streptomycin resistance gene over here shows you that any cell that gains this particular plasmid will also gain resistance to streptomycin and ampicillin. Now down here, this lower right looks really complicated, partly because it's showing you all of the places that you can cut this plasmid so as to modify it. Many plasmids are very much man-made structures. They are, they're like, you know, building Legos, if you will. They're things that researchers can put together. They can cut them where they want to cut them and put genes in where they want to put genes in. And so all these things that say SMA1 and XMA1 and EchoR1 are showing you where could you cut the plasmid if you wanted to make it, you know, if you wanted to knit together a new plasmid. So this is very much about how you could modify the plasmid. But notice that this plasmid also has chloramphenicol resistance. That's an antibiotic. Ampicillin resistance. So this is another resistance plasmid, another R plasmid. What is the scary thing about a plasmid in which you might have this plasmid that has on it, say, this ampicillin resistance gene, kind of like what we see in this plasmid here. So we'll just say that this is AMP resistance. Sometimes we do a shorthand like that, AMP resistance. And then what if we have on this also another gene right here, and this gene encodes for resistance to Clorox wet wipes. So how can we write this in shorthand? We can say Clorox wet wipes, CWW, resistance. So resistance to Clorox wet wipes. I want you to tell me what is so crazy about thinking about a plasmid that has a gene for Clorox wet wipes resistance and a gene for ampicillin resistance, and what would be kind of crazy about that particular plasmid regarding selective pressures. Okay, so relate it to that idea that we've talked about already of selective pressure, and tell me the horror story that could ensue there. The last thing that I want to talk about in today's vodcast is actually to look at Norbert's cell wall. So most bacteria and archaea have a cell wall, a rigid layer that is external to the cytoplasmic membrane. So here on our model, you can see that the green layer actually indicates the cell wall. So remember that the yellow is the cytoplasmic membrane dictated by the fluid mosaic model, and then we have the green layer, which is going to be the cell wall. But we can't really tell what kind of cell wall this is just based on that green layer. And so what I want to show you is that depending on the type of bacterium, the cell wall will differ. Now, this is a point of divergence between archaea and bacteria. Only bacteria have this molecule called peptidoglycan that serves as rigidity in the cell wall. So for a moment, we're going to make the assumption that Norbert is a bacterium. We've been making that assumption all the way along. So we're going to say that there are two types of cell wall that Norbert might have. And I'm going to show you a model that shows these two types, these two possible types of cell wall. And I've got lots of models of these types of cell cell wall. But this particular cell wall right here, so I want to make sure everybody's with me. Now, if we were to zoom in, right? Okay, so if we were to zoom in to that green layer, and we can look at the ultrastructure of that green layer on the cell wall, we would see either one of two things. We would either see a gram-negative cell wall, and that's what this is here. So in a gram-negative cell wall, we have a cytoplasmic membrane, and that's going to be true no matter what. Then we have this region called the periplasmic space, and then we have an outer membrane. So the outer membrane is typical of a gram-negative cell, and in that outer membrane, there are little molecules called lipopolysaccharide here that this person has shown with the little squares of orange. So this is an outer membrane, a periplasmic space, and an inner membrane. And inside of the periplasmic space is something called peptidoglycan. And this is the molecule that I want to start our conversation of the cell wall on. Now, on the other side here, we have another type of cell wall. If we were to zoom into Norbert and see this kind of cell wall, we would say that Norbert is a gram-positive cell. In a gram-positive cell, of course, we still have the cytoplasmic membrane, but then we have this super thick layer of many sheets of peptidoglycan. Up to 30 of them would typify a gram-positive cell. And then we have these little molecules called teichoic acids that are indicative of a gram-positive. So if we zoom in and we see this, we say that Norbert is a gram-positive cell. If we zoom in and we see this, we see that Norbert is a gram-negative cell. But notice that what is conserved in both bacterial types of cell walls is that really cool looking peptidoglycan. So peptidoglycan is found in all kinds of bacterial cell walls. Now, quick caveat, not all bacteria have cell walls. In fact, there are some bacteria that don't have them at all. And in fact, if you ever see the term mycoplasma, anything that has a plasm or plasma, you can look at that and say, hey, that's a bacterium that doesn't have a cell wall at all. So it's an exception to the rule. In fact, some of you have met this one. It causes walking pneumonia. So if you've ever suffered from that, you've had Mycoplasma pneumoniae. But here we see that this would be an exception to the rule. Most bacteria have a cell wall. Mycoplasma does not. So let's zoom in and look at the ultrastructure of peptidoglycan. I was thinking, what can I use as an analogy for peptidoglycan? Because peptidoglycan or murine is a very rigid structure, but it's also very, very permeable. So it adds a lot of rigidity to the cell wall, but it does not keep substances from diffusing in and out. That is, it's not like a cytoplasmic membrane. It's not a bouncer at the bar. It's instead more of a rigid layer that provides structure but allows porosity. So I was like, what could I think of that would give you that idea? And I found a tea strainer in my cupboard, and a tea strainer is a good example of what we could think of as peptidoglycan, where it's very rigid, right? This is very rigid, but it also is very permeable, right? We know that it allows stuff to diffuse in and out of those pores on the pores in the tea for the tea to strain through. So this is a very good analogy for what we would think of as the role that peptidoglycan plays in a cell wall. So you're thinking, well, molecularly, what is it made up of? Well, it's actually made up of derivatives of sugars called NAM and NAG. This looks like glucose, doesn't it? That six-membered ring. And so it is very similar. In fact, this is N-acetyl glucosamine. That's what NAG stands for. And N-acetyl muramic acid. So they are derivatives of sugars that are linked together very much in the way that that carbohydrates are. So this is a repeating polymer of NAM and NAG and NAM and NAG and NAM and NAG, or N-acetylmuramic acid and N-acetylglucosamine. They're in a long polymer. So that long polymer would continue all the way around the cell. So in fact, if you were to look back at our model of the gram-positive cell wall, if we were drawing the entire cell, we would see this thick layer of peptidoglycan. So if this was NAM, NAG, right, that would proceed all the way around the cell. That is, it would be all the way around Norbert, right? It would It'd be nam, nang, nam, nang, nam, nang, nam, nang, and many, many layers all the way around there. Okay, so let's look in at the thing that knits together the nam and the nag. So here's the nam and the nag, and here we're just showing them by name rather than by their structure. And then notice that attached to the nam residues are amino acids. They're amino acids like L-alanine, D-glutamate, D-alanine, and it forms a chain of 4 of them. So because there are 4 amino acids, we call this a tetrapeptide chain. So we see a tetrapeptide chain attached onto the NAM residues. And then the tetrapeptide chain is linked to another tetrapeptide chain on another NAM-NAG polymer. So this is actually what you would think about as if you crochet, right? This is kind of pulling together the NAM and NAG chains to enable them to get linked to one another. Now, depending upon the bacterial cell wall, they may be directly linked. That is, one amino acid in the tetrapeptide chain may be linked directly to another, or they may be linked via what's called a glycine interbridge. So this glycine interbridge is more common in gram-positive bacteria. So in our model, again, and you guys are so laughing. In our model, we can see that this gram-positive cell would have, and we'd have to zoom in. We can't see it on this model. We'd have to zoom in there to see the tetrapeptide chain and the interpeptide bridge. But this gram-positive cell would have the interpeptide bridge linking together the tetrapeptide chains, whereas we would be less likely to see that in the thin peptidoglycan of a gram-negative cell wall. But I do want to make note that this tetrapeptide chain is really weird. That is, we think about the 20 standard amino acids that make up a protein. Well, these are none of those really, excepting L-alanine. L-alanine is, but D-glutamate, D-alanine, these are unusual. These are weird. In fact, mesodiaminopimealic acid, have you ever even heard of it? It's nuts. It's not one of the 20 standard amino acids. Bacteria do this on purpose because they don't want their tetrapeptide chain to be degraded readily by the normal proteases or enzymes that digest proteins. They want them to be protected from that. So that's why they make up their tetrapeptide chain out of really weird amino acids.